Welcome back, everyone. We'll be continuing this afternoon with our second panel, which will be exploring the intersection between the environment and AMR. We'll be hearing today from Dr. Jim Stack, a professor at Kansas State University, Department of Plant Pathology, and Dr. Nancy Chow, the Data and Quality Team Lead from the Mycotic Diseases Branch, CDC. Welcome. Dr. Jay Garland, the Senior Research Scientist at the Center for Environmental Solutions and Emergency Response, the EPA. Welcome. And the last presentation will be joining us virtually, Ms. Mariah Ewell, the Program Director for International Activities at the National Science Foundation. So welcome, everyone. Thanks for sharing your expertise. And Dr. Stack, if you'd take it away.
Good afternoon, and thank you for the opportunity to contribute to this discussion. I was asked to provide a plant health perspective on the impacts that climate are having on food systems, with particular reference to antimicrobial resistance. Next slide, please. So the impacts of climate on plant systems is pretty clear. We're seeing migrations of whole plant communities to higher elevations. We're seeing migrations to more northern latitudes in the northern hemisphere. The problem is the species aren't migrating all at the same rate, and so we're seeing separation of some plant species from their pollinators. Altered flowering times has been reported around the planet in nature with earlier flowering by 10 to 14 days in some areas. Again, this is providing the opportunity for a temporal separation of plants from their pollinators. We're anticipating and might actually be starting to see increased disease prevalence and severity in some of these areas, and the impacts are not uniform. It's not very even isobars with the latitudes. It's more of a mosaic of the impacts. Next slide, please. Impacts on the pathogens are similar. We're seeing geographic range expansion in some pathogens and their vectors, and that overlapping ranges might provide for the opportunity of acquisition of new vectors, which then could expand the host range of some of these pathogens. A recent report has indicated that the climate change might actually be selecting for pathogens with greater dispersal capabilities. The emergence of hybrid species we're experiencing not necessarily as a consequence of climate change, but as a consequence of global trade. And the challenge with that is that the hybrid species has a phenotype not predictable from the parental types. So we're seeing hybrid species that have a different host range and different virulence patterns than either parent. And then there have been some reports about seeds and bacteria that have been frozen in permafrost for 30,000 years that upon melting, they are fully viable. The consequences of that are hard to predict. Next slide, please. So climate cannot be considered in the absence of all these other challenges to plant health that impact human health and well-being. These challenges must be addressed as systems, as hard as that might be. If we address them individually, we will fail collectively, and that goes for antimicrobial resistance as well. Population, demographics, human behavior put increasing pressures on each of these challenges. Next slide, please. Currently, Africa and Asia house approximately 75% of the world's population. This is on land that in independent assessments have determined that over 40% of the land in these regions is already seriously degraded. We're going to add over 2 billion more people to the planet in the next 30 years, many of them in this region that are already experiencing chronic hunger and malnutrition. Next slide, please. The consequences of climate include chronic crop loss or failures, outright failures, that are going to maintain the undernourishment that we're experiencing already in this region, that leads to low immune function, that leads to increased susceptibility to infections, including foodborne pathogens. Increased use of antibiotics is common in response to the malnourishment, and we're seeing widespread overuse. You can go into the corner store and find these products on the shelf and buy them without any prescription. A challenge anywhere is a challenge everywhere. Next slide, please. So this is just a collection of manuscripts to indicate that what I'm going to tell you is not just a one-off, that this is very well documented now in reputable journals, including Cell, Frontiers in Microbiology, and Applied Environmental Microbiology, and others. That our concept that these foodborne pathogens are surface contaminants of plants is incorrect. These are full-born colonists of plants. A study, that lower one on the right, the Salmonella, looked at gene expression, and they determined that 30% of the genes that are upregulated in the intestine when Salmonella infects are the same 30 genes upregulated in Salmonella when they infect plant tissues. So this is a very serious issue for us to take into account. Next slide, please. That upper image on the left is E. coli colonizing a plant through a natural opening, a stomate. The image on the bottom is subsequent systemic colonization of that plant in the vascular system. When these foodborne pathogens are co-inoculated with a plant pathogenic bacterium, we're seeing 100 to 1,000-fold increase in cell numbers in these plants. So clearly there's a cooperation between foodborne pathogens and plant pathogenic bacteria. Is there meaningful crosstalk? The evidence would suggest yet. Recently, Salmonella plasmids have been detected in 2 different genera of plant pathogenic bacteria. We've found vancomycin resistance genomic island in another plant pathogen, Rathibacter. Rathibacter is commonly consumed by livestock in colonized grasses. Multiple antibiotics are used to manage diseases in plants. And resistance has been detected in plant systems. So one of the consequences of this increasing relationship between foodborne pathogens and plants might be plants becoming a more efficient vector of moving antimicrobial resistance around the planet. We now have seen different antibiotic resistant strains popping up in different plant products. So the fact that we are seeing the genomic islands being transferred from foodborne pathogens into plant pathogens suggests that there's some level of exchange of genetic information. So are foodborne pathogenic bacteria evolving to become bona fide plant pathogens? And then the converse is true. Are plant pathogens, is it something we should anticipate that plant pathogens are going to ultimately evolve to become human pathogens? These are hypotheses that are actually being tested. Next slide, please. Fungicides are one of the main tools in the toolbox for managing plant diseases. Fungicide resistance is becoming a major problem. Many of our fungal species are hypervariable. They undergo constant recombination, making host plant resistance unstable. Wheat is the second most important staple food crop globally, grown on more acres globally, providing 20% of the calories and 17% of the protein in the human diet. A disease emerged in South America in the 1980s called wheat blast. It's a seriously damaging disease, oftentimes resulting in no yield in a field. Well, it was confined to South America until 2016 when a major outbreak occurred, major epidemic in Bangladesh. So it's now present and spreading in South Asia. And then in 2017, the same fungus is present now in Tanzania. So now gained a foothold in the Eastern Africa wheat belt. The challenge we have right now is it's hypervariable and fungicides are the only viable management strategy. But in Brazil now, we have populations of this pathogen with simultaneous resistance to 3 different fungicide chemistries. Next slide, please. In October of 2022, the World Health Organization published their priority list of fungi. And in that statement, they indicated that their records indicate 1.7 million deaths annually from fungal infections, now more than malaria. Fungicide resistance is a major threat to effective treatment of fungal infections in humans. We heard earlier about the Aspergillus fumigatus. I will stress that that species is not the only species of Aspergillus that is of both plant and human health concern. Primary fungicide therapeutics used for humans are the same chemistries used for managing fungal diseases in agriculture and horticulture. Those fungicide-resistant Aspergillus fumigatus strains actually arrived from an agricultural environment. Now, a recent statement or a publication was mentioning this new chemistry, olorifim, promising new chemistry for human infections, but now a label is being pursued for agricultural use. Is this rational? Next slide, please. As new pathogen strains emerge due to climate change, and antibiotic-resistant and fungicide-resistant strains emerge due to public health and plant health practices, we will ensure their rapid dissemination around the planet through global trade and travel networks. It's the way we do business. If you look at that lower image on the right, on the left, each one of those little airplane icons is an actual flight in progress at 8 p.m. at night, every night. That's what the world looks like in the skies. The image on the right indicates the traffic across our oceans is about the same. The center image is the global trade network for cereals. Every commodity is like that. We move enormous amounts of people and product around the planet in short periods of time. Next slide, please. The World Food Program says hunger is tied to civil unrest. When people get hungry enough, they move, and populations on the move are prone to conflict. The World Food Program provides emergency food aid to over 100 million people every single year. During the COVID era, it jacked up to almost 200 million people per year. Reuters published a paper, Climate Conflict Set to Plunge Millions into a Food Crisis. Take a look at those lower images. In 2008, we had food riots in over 30 countries, resulting in the overthrow of 2 governments. The center image, Tunisia, 2010. Although there were many underlying issues, it was the dramatic increase in the price of wheat, the price of bread that drove the people into the street for those protests in Tunisia that spread across northern Africa into the Middle East, the repercussions of which we're still facing today. Last year, no different. Food riots as prices get out of control. So as we face climate-induced stress that impairs our ability to produce food, and we add 2 billion more people to the planet, we should anticipate some challenges. Next slide, please. Policy provides the framework for decision-making. We need coordinated policies for strategic deployment of antibiotic and antifungal chemistries across application sectors, namely public health and agriculture. This needs to be an international collaborative process. A challenge anywhere is a challenge everywhere. These challenges must be addressed as systems. If we address them individually, we will fail collectively. If we don't get the policy right and manage the antibiotic and antifungal chemistries rationally, we will lack the tools necessary to feed 10.5 billion people and the therapeutics necessary to protect them from bacterial and fungal diseases. Getting it right matters. Next slide. Thank you.
Thank you, Dr. Stack. Dr. Chow, if you'd take it away, please. Welcome.
Thank you. Good afternoon, everyone, council, and those online.
Could you pull your mic just a little closer,
a little closer?
please? Yeah.
Okay.
Thank you.
So I'm going to share 3 examples of how environmental changes can impact fungal infections in AMR. Next slide, please. So the first thing to point out there is that fungi are all around us, and disease-causing fungi are all around us in the environment. They are inextricably linked to our environment. And so environmental changes, even small environmental changes, can dramatically affect how fungi evolve, which fungi grow, where they grow, and whether they're able to affect humans and other animals. Next slide. And when we're thinking specifically about rising temperatures, it's interesting to note that few fungi can grow at the human body temp of 98.6 Fahrenheit. And so when we're considering all of the fungal species, and it's estimated to be quite a bit, about 2 million species, it's only a few hundred that can survive at these high temperatures. And one of the theories that exists is that humans have evolved to be able to have such a high body temperature as a protective mechanism. And so if we take that theory one step further in the context of Earth's rising temperatures, the question is, will there start to be more fungi that adapt faster than us to increasing human temperatures, and will they kind of fall within this few hundred category and be able to go on and infect humans and cause disease? Next slide. Well, if you ask HBO, they're going to tell you yes. You know, just couldn't resist putting this in here. I'm thinking that some of us, many of us, may have seen or even at least heard of this new HBO series called The Last of Us. And it's based on this very premise that a fungal pandemic occurs in response to increasing temperatures, and it causes a global zombie apocalypse. Now, while unrealistic, this has been a wonderful opportunity for CDC and others to communicate to the public about fungal infections. And here you can see an op-ed written by our branch chief, Tom Chiller, in response to the show, telling us about fungi that we should be thinking about. And here, again, I'm going to expand on 3 of these examples. Next slide. So the first one is valley fever. This is an infection also known as coccidioidomycosis. It's quite a handful. It is caused by 2 species, and here you're looking at one, which is Coccidioides immitis. Next slide. And these species, Coccidioides, they're dimorphic in nature, so they predominantly exist as 2 forms. The first form, the environmental, is how it grows in the soil. It grows as a mold, and as it's reproducing, it can form what are called arthroconidia. And these arthroconidia can become airborne, and if they're inhaled, it converts to the host-associated form. It can survive at the human body temperature, progress through its life cycle, and this can result in a symptomatic pulmonary infection. And if it's not treated properly, this can also progress to a disseminated severe infection. Next slide. And what we're finding from much of our enhanced surveillance data that we've collected over the years is that most patients with valley fever, they're diagnosed incorrectly. And most of these are prescribed multiple, not one, multiple courses of antibiotics when they should be receiving antifungals. And of course, we know that this overuse and misuse of antibiotics helps to promote antimicrobial resistance. And so in response to this, CDC, one of the big things that we try to do to try to, increase more rapid and accurate diagnoses of valley fever infection, next slide, is to better understand where these species live. And so if we're able to look at these areas of endemicity and map them on, and this is looking at skin testing data, case data, soil sampling, you can see that these areas of endemicity, what was projected in the 1950s, and then currently, it's predominantly in the southwestern United States with pockets as far north as Washington State. Next slide. But if we model this out, and we use climate projections from the years 2035 and 2095, we see that this geographic range is expected to expand in the US. And so incorporating rising temperatures, changing precipitation patterns in the context of how we know cocci likes to thrive, which is predominantly very wet winters followed by very dry, arid summers. What we're seeing is an expansion that more than doubles. It extends northward, and the number of affected states would go from 12 to 17. So very dramatic effects. Next slide. And so in combination with this idea of the geographic expanding range with an increase in extreme climate events where it can aerosolize these arthroconidia, this is going to lead to more people at risk for valley fever. And so overall, valley fever is an example of how environmental changes can expand where pathogenic fungus grows and ultimately putting more people at risk for these infections. Next slide. So the next one is aspergillosis, and I was just joking with Dr. Stack here that he's stolen a little of my fungal thunder, but repetition never hurts. So aspergillosis, it can be an invasive mold infection, especially among those who are immunocompromised, caused by Aspergillus species, and the primary causative agent is Aspergillus fumigatus. Next slide. And what we're seeing is that agricultural fungicides that are similar in chemical composition, sharing the same mechanism of action as clinical antifungals that we're using to treat patients. The use of these fungicides, it's driving drug-resistant Aspergillus fumigatus infections, and it's all related. We've heard about climate effects on crop pests and how this can drive our need and use for more fungicides. And in the case of triazole fungicides, this can induce the development of azole-resistant A. fumigatus strains in the environment that can then go on to cause infection. Next slide. And, when we first started hearing more and more reports of azole-resistant A. fumigatus infections, it wasn't entirely surprising, just because since the 90s, we've seen these infections, especially among people who are receiving long-term azole therapy for chronic pulmonary aspergillosis. But it was when we started to see it more in people with no prior azole therapy. So they hadn't received these drugs before. And that kind of cued us into this potential link with fungicide use. And fortunately, we're able to track strains of azole-resistant A. fumigatus that are specifically linked to fungicide use through genetics. So there's two genotypes, we've heard of these, TR34 and TR46, that are specifically linked to fungicide use that we can use for some of our surveillance efforts. Next slide. And, of more recent, what we're finding as an additional concern to this issue is that the promising new clinical antifungal that's been shown to be effective against azole-resistant aspergillosis, it shares a mechanism of action with a recently authorized agricultural fungicide. So this new clinical antifungal, Olorifem, it's the first member of a new class of antifungals being used to treat invasive mold infections, and it's been found to be effective against azole-resistant A. fumigatus, which is exciting. But in parallel, a new recently authorized, as of last year, fungicide called iplufenequin, it is authorized for use and shares the same mechanism of action for Olorifem. So, it raises the concern, are we just heading down the same path? Are we repeating history of what we're seeing with azoles, where we're using that same mechanism of action for agricultural fungicides as what we're using for clinical antifungals? Next slide. And, it's important that we address this issue. So what's been very exciting is that we've created an interagency working group with CDC, EPA, USDA, and others to try to address this issue, because this is a global One Health AMR threat. Everything that we've seen right now in the US, it has precedence. It was first reported in Europe. When we look at our average agricultural triazole fungicide use, we know that over the years it's increased dramatically in the last 10 to 15 years. And so we have to continue to monitor and conduct surveillance on this issue. And critically, we have to try to preserve the efficacy of new antifungals coming down the pipeline. Next slide. Okay, so the last example to share is about Candida auris, C. auris, and then next slide, thanks. And I know that we've heard a lot about this fungal superbug, especially it's been in the news a lot this past week. So just as a quick recap, C. auris, it emerged suddenly about 15 years ago, 3 separate continents independently. It causes outbreaks of invasive infections in healthcare facilities. It's been reported in over 40 countries, and in the US, it's been reported in over 25 states. Highly transmissible HAI, where it colonizes the skin. We see person-to-person spread. It's highly resistant to antifungal drugs. It's pretty much intrinsically resistant to azoles. And there are increasing rates of pan-resistance, where we see resistance to all 3 available classes of antifungals for invasive infections. Next slide. And so one of the unknowns that still exists is, how did this happen? How did C. auris just emerge suddenly on 3 continents with very distinct genetic strains. We know it's independent. And one of the theories proposed by Arturo Casadevall and colleagues suggests that C. auris emerged because of environmental changes. So it proposes that these rising ambient climate temperatures, it allows for selection for fungal species that can grow and reproduce at basal body temperatures of humans and other animals like birds. It further proposes that C. auris previously existed as a plant saprophyte, so living off of decaying plant material in the wetland ecosystem. And it was there that it was able to adapt to high temperatures, high salinity environments, and that there was some sort of intermediate host that was intermediate between the wetland ecosystem and humans. And this, likely an animal, could have been a bird, but it helped to promote, drive migration, as well as increased human contact to then enter into the human population as a skin colonizer, and then eventually into the healthcare environment where it can cause invasive infections. Next slide. Well, one of the limitations to this theory when it came out at first was that C. auris just hadn't been isolated from the natural environment. But very recently, it was isolated off of the coastal wetlands in the Andaman Islands in the Indian Ocean. So it's a data point that further supports this theory. But the question is still out there. It's speculation, and it's still unknown. Next slide. How a species like this emerges. But if you're in the camp that environmental changes plays a substantial role, should we start to anticipate the occurrence of more pathogens like C. auris? And then last slide. So some key takeaways: most disease-causing or pathogenic fungi, they originate from the environment. Environmental changes can affect what pathogenic fungi emerge, where they spread, and what fungi dominate. And then increasing and inappropriate use of environmental azoles like fungicides will lead to more drug-resistant fungal pathogens that can affect humans. And then I'll just close out by saying, think fungus, save lives, which is the really last slide.
Thank you very much. Dr. Garland?
Thank you. What I want to talk to you today about is several initiatives within the EPA Office of Research and Development. And I guess the 2 points I want to make here is that, number one, I am with the Office of Research and Development, not with the Office of Water or the Office of Air. The initiatives here are research-based, not regulatory-based yet. We do research to inform eventual regulation and those kind of policy decisions. Secondly, it is initiatives that we are involved in, but one of these initiatives in particular, in terms of monitoring, heavily leverages other people, next slide, other interagency efforts. This has been done as part of the NARMS program. So this is a collaboration with FDA, USDA, and CDC. I particularly want to call out FDA. Collaboration is one thing. Transferring money between agencies is quite another, and they have transferred money to EPA to enable this research. So there's a strong commitment there for the environmental component. And also I want to call out USDA, their involvement. The ARS staff really worked very diligently as part of this, actively developing methods even during the COVID period. Next slide. So the 3 things I want to talk about is, one, this interagency collaboration on developing a surface water pilot as part of NARMS. That'll be most of the talk, but I also want to talk about an upcoming RFA that we have in place to really look at the fate and transport of AMR through wastewater treatment, human wastewater treatment plants, as well as the relative impacts of different sources on the environment to understand, in fact, what is the relative impact of wastewater, human wastewater, which is one of our regulatory responsibilities. And just to note, that second bullet, it's not about looking at wastewater reflecting back on population levels. Wastewater surveillance through COVID and the important work that CDC is doing is different in this sense. We're looking at not reflecting back on population levels, but really looking at what is the transport and the environmental exposures that are occurring from that. And then lastly, I want to talk about risk models and how all of this really needs to be translated into risk. The question this morning about hazard versus risk, couldn't agree more that this data is only valuable if we can fit it into effective risk models. Next slide. So there's been a lot of discussion over the last decade or so about environment, the role of the environment. There's lots of review articles. This one is from Larson et al., one of the leaders in this area. And, there's three major efforts trying to understand what the relative contribution is from different sources in the environment is a reoccurring theme. Secondly, understanding the role of the environment on evolution of resistance is also important. And then also this third bullet is that risk factor, translating any of those measurements into understanding what risk is important. And then finally, what do you do with that information? You look at efficacy and feasibility of interventions. Next slide. This slide has a couple bullets in it. The top one is yet another review from 2018 that specifically talks about surface water and the factors that you need to consider with surface water. It shares some of the same considerations. One, looking at spatial distribution to inform risk and looking at sources and selective pressures for amplification and transmission. And then also this third element of really defining and standardizing sampling analysis methods, really critical. There's no lack of data, but there is a lack of consistency in the data that's produced in the environment. That is work on the environment, on water, and I show that because this first initiative is through the NARMS program, the NARMS strategic plan, in response to input from their own SAB that really you need to kind of walk the walk of One Health. Where is your environmental component? They really reached out in collaboration with us and the other agencies to try to develop a pilot effort in the environment. We ended up selecting water as that initial pilot project, not because that's the only environmental component that's important, but it's an important confluence and integrator of impacts in a watershed, so it was a good starting point. Next slide. And so beginning, actually quite overlapping very nicely with the pandemic, we formed this surface water pilot team, this interagency team, and developed our objectives. Obviously, it's to develop this pilot effort within the One Health focused NARMS, but below that, really, the goal was to develop a national scale quantitative assessment of AMR and surface water. And quantitative because we wanted to have a standardized measure and maybe a library of samples that we could look at long-term trends. That's one sub-objective. We also wanted to develop quantitative data that we could feed into risk models. And third, get at this, help understand drivers of occurrence and selective pressures. That third bullet, some of it clearly is going to be focused, targeted studies that are more mechanistic in nature, maybe not a long-term monitoring program, but we wanted to develop the monitoring program to help inform that. And then finally, all of this, again, overlapping with trying to use this information to inform and understand what critical control points are and what current and new mitigation strategies are having on AMR and the environment. Next slide. So when you're designing this kind of study, I guess one really maybe crude way to phrase it initially is, do you want to go big and slow, or small and fast? And I guess when I'm talking about slow and big, it's like the spatial scale of it. Do you want to get a national scale picture of this? And that picture on the top is a national scale probabilistic survey of all the rivers and streams in the U.S. that EPA conducts on a 5-year basis. I'm going to talk more about that as a way to go big and slow. It gives you that national picture, but you only do it every 5 years, so it's slow. We know that there are factors that drive levels that occur at much higher frequency, like rainfall events. The data on the bottom is from a CDC preliminary work in the Chattahoochee River, where they can link, the levels of AMR in river water to rainfall events, which is not surprising. So do you go big and slow and small and fast? And our decision, next slide, was to try to do both. And the phase design of this effort, the first phase was to initial testing of the methods and standardization of methodologies, going back to the point about environmental work really needing to incorporate that. And that work was done over the last couple of years, and that's in place. And I have to say that we collaborated quite extensively with work that Dr. Pruden, who you met earlier, was doing as part of a WRF grant that she had, the Water Research Foundation grant, to develop standardized methods for water and wastewater. We're using the same methods, and I'm going to describe that in the next one of the next slides. But then the second phase was to implement this watershed-based study with the idea that that's going to give you more mechanistic understanding at a finer scale. And we've initiated that and are in the middle of that. The third phase is then this larger, big and slow national probabilistic design using the NRSA, the National Rivers and Streams Assessment, which I'm going to mention. And that's the pilot effort, those first three. And then the idea is phase 4 would be beyond the pilot. What are you going to do really long term? And that's the idea is the pilot would inform that. Next slide. So the analytical targets, the methodologies. The question earlier today, someone asked, do you go culture, you go genomics? Well, again, we kind of said you need to do both for different reasons. You need to do culture because it gives you quantitative estimates of pathogen levels that you may use for these risk models. We picked enterococci and E. coli because they are readily used quite extensively in aquatic systems as other indicators of fecal pollution, so they could be adapted for this use. We also use salmonella, something that's not typically used a lot in water work, but it was linking back to the food cycle and the NARMS effort. We also did targeted gene analysis because, that's limited in nature. The culture work only allows you to look at several targets, but the gene analysis, you can look at up to 100. That's our plan of ARGs. And in addition to that, look at fecal source trackers so we can relate the antimicrobial resistance to different sources of fecal pollution in the environment. And then finally, looking at metagenomics as well to get a broader view of the resistome in surface water, maybe define improved targets for further work on targeted gene analysis and maybe different criteria. So next slide. Just a little more detail on that. In terms of E. coli, specific method that we're using there, we're looking at both the susceptible and cefotaxime resistant, so ESBL E. coli here. We're generating that data on numbers and then also transferring the isolates from that to our colleagues in FDA for whole genome sequencing to link back to identification of those strains to other sources where they're seeing it in the food cycle. Enterococci, we're doing total enterococci, but also vancomycin resistant enterococci. Again, looking at both the susceptible and resistant and doing that in a quantitative way. For salmonella, we're not doing it quantitative, we're doing an enrichment stage to see a presence, absence of salmonella, and then also doing whole genome sequencing of those. Next slide. And then in terms of the watershed study, these next couple slides, just going to pass over them briefly. If you want more detail, we picked a watershed where we had an existing water quality monitoring program in place. And this is, the point made earlier by Dr. Fukada about linking to existing work. This is a watershed-based effort that is looking at nutrient management and nutrient pollution in the watershed, so we could leverage that existing framework that was in place to look at AMR in that watershed. And it's a gradient of urban to rural, including a lot of agriculture, not necessarily a lot of animal ag or animal fecal use in that, but there is recreational exposures, a reservoir in there, so there's a lot of interesting attributes to that watershed, not too dissimilar than a lot in the Midwest. Next slide. We're looking at the watershed study because it complements the larger study, which we're going to talk about next, to get more at the seasonal variation and the specific drivers in the watershed. Next slide. But let's talk a little bit about this national scale, because I think that's a little bit more unique than maybe what you've seen before in terms of more targeted studies. So the NARS program, the National Aquatic Resource Survey, is something that EPA does on a 5-year cycle. We cycle through national rivers and streams, coastal condition assessment, and then lakes and wetlands. We do that on a 5-year cycle. The graphs on the right, for the rivers and streams, the study is structured around ecoregions. If you're going to study the rivers and streams or any ecosystem, you need to look at what the differences are just based on climate and in ecology. So this study is a probabilistic design of all of the rivers and streams in the U.S., and it's structured around ecoregions. And we'll go more into details about that with some of the data that we generated. So next slide. This is a kind of a blow up of that map of the rivers and streams. There's some of the names here, Southern Plains, Northern Plains, xeric, mountainous, west. These are all things that you can look at this slide and memorize at your own interest. But the point is that there are these different districts, and the study is structured so that these sample points are selected to represent certain rivers and streams. They're not meant to target certain specific sites, say around a city or a given farm, but they are meant and designed to represent all of the river stream miles in the U.S. because the goal of this program originally was to look at impairment so that we as EPA could map out the aquatic resources of the U.S. and say what's impaired and what isn't. And we do a lot of different measurements on this, including water quality, fish, macroinvertebrates, algae, all kinds of measurements. We also do fecal indicators, which are criteria for impairment. But in addition to that, we built off of that to look at at AMR within these systems. Next slide. Now, I'm going to talk a little bit about a pre-pilot we did, and this is some work that we shared with you probably 4 years ago, some of this in early format with the PACCARB. And this pre-pilot was done so that we were trying to test out and see if this was valuable as a way to look at national scale patterns of AMR. And I'm showing this so that you can understand that in the future, all those additional analytes that I mentioned, the culture, the 100 ARGs and the metagenomics, for this pre-pilot is just these relatively small number of ARG targets. But the way that we analyze the data for this study would be the same for the larger scale study that we're doing now, the pilot study, but just much more information, many more analytes. Next slide. And so the results here, these maps, maybe it's hard to see from where you're looking at, but what you can generate based on the data is national maps of the levels of ARG, and you can classify it out overall in the US, but also by ecoregion. So that's what you see here is this is a heat map of ARGs from that pre-pilot throughout the US. And in this case, darker blue means cooler or actually lower levels, where the lighter colors means more. And you can see a couple things. One, overall, of the ones that we looked at, SOL1 and INTI1, which is not, it's the not necessarily a direct resistance gene, but indicator of gene exchange. Oops, sorry. You go back one slide. What you can see is that there's this a gradient of east-west a little bit, east of the Mississippi, west of the Mississippi, but there also are specific differences you can see between ecoregions. And the fact is, we really could observe these national scale geospatial patterns based on, which is kind of striking because going in, we didn't know whether we'd see that because these are a single sample taken from a watershed over the course of once a year during low flow conditions, in those rivers and streams, base flow conditions in those rivers and streams. So some of the details there you can look at as your leisure. Next slide. The other key thing about any environmental work with AMR is trying to understand what the level is versus kind of baseline analysis. Because since this isn't a normal environmental contaminant, trying to understand, have reference or control sites is very important. This study does allow us to do that because within all those ecoregions, we have sites that are classified either as good condition or poor condition and intermediate. Actually, I'm just showing good and bad here, or good and poor, based on other water quality measurements that we take. And those are listed on the right-hand side. So next slide. What that allows us to do is look at any of these response variables. Here it's these ARG levels, and look at what their level is in the reference sites or least disturbed versus those most disturbed sites. And clearly you can see that disturbance in the overall site does reflect an increase in ARGs. Next slide. Next slide. So the general conclusions from that study is that the ARGs did show this, again, the significant geospatial pattern at a national scale. They were good quality rivers and streams, had lower ARGs. So, I mean, if you want to control ARGs, have clean streams. Now, that's a little bit, off the cuff, but the point is the fact that it's a good quality stream, the fact that it's not getting those inputs, partly, that's probably it, but also is it the ecological integrity itself that may be driving something about the loading and reservoir of ARGs in that site. The INT1 really was a strong operational ecological indicator, condition indicator. That's something that we're very interested in. It's part of the study. It's having indicators. And then specifically for some of these, we saw relationship to urbanization in the watershed. We know other information about that watershed, the level of urbanization, the level of watershed integrity based on other measurements we do in the functionality of those watersheds. So we can see linkage of the levels to these other indicators within the watershed. So this formed the basis for really working with FDA on building that study into the ongoing NARMS program in 23 and 24. So next slide. I just want to touch on other thing real briefly. One is the National Academy of Science report that came out last year among the many things it talked about, it covered the need for looking at the effluent and discharges from human wastewater as this important conduit between human activities and the environment. And they specifically recommended that EPA should provide guidance and resources to states for testing point source discharges at their water treatment plants. We take that recommendation very seriously. We're not at the point now of providing resources and recommendations directly to them to do that, but we have identified a national priorities funding to look at that antimicrobial resistance in wastewater and sewage sludge treatment and the relative impacts, as mentioned earlier, in the environment. That's going to be a RFA that's coming out this year and would be to probably be funding levels between $9 and $10 million. So very significant investment for us to really generate information to better understand this issue of wastewater, human wastewater impacts on the environment relative to others. Then finally, just in closing, real quickly, I just want to go back to this idea of risk models. We are actively working on risk models. Dr. Pruden mentioned this morning that there is the standard framework of quantitative microbial risk assessment, of looking at hazard identification, exposure assessment, dose response, then getting at the actual risk characterization. That's a very standard approach that we use in our regulation for drinking water, recreational water use. The point of these slides, I know there's a lot of detail here. We are modifying that in collaboration. This is work done in collaboration with Carrie Hamilton from Arizona State University to modify that approach to utilize it for AMR. AMR is much more complex because you have the movement of the genetic material. You have differences in dose response, differences in the genetic exchange, and even differences in the impact of a given infection. Next slide. We are this overall framework is being developed, and next slide to close, we are utilizing that framework for specific questions related to potential regulatory or actions. In this case, we looked at the question of MRSA infections versus staph infections. If you're going to take wastewater and reuse it for showering, and that's a reuse option that's on the table in certain communities in the U.S., and so we're looking at that and understanding the relative impact of just the Staph aureus infection versus MRSA infection, including understanding horizontal gene transfer rates during treatment and also the differences in dose response between those different strains. So just as a broad overview, this is emerging work, and it's something that we're committed to doing to improve these models so that we can transfer that hazard assessment to real risk assessment, especially for waterborne exposures. This is for a reuse option. We're doing it for ESBL E. coli for recreational exposures. And as I mentioned at the very first slide, we are going to try to work with our office, not of Office of Water, but Office of Pesticide Programs, to develop a more quantitative risk assessment framework for the use of antimicrobials in ag, which the Office of Science or Office of Pesticide Programs regulates. Thank you.
Thank you. I believe we have Ms. UL with us online. If you'd go ahead and take it away.
Great, thank you very much. So I think that my fellow panel members have outlined fairly clearly the challenges of AMR. So really what my presentation is going to focus on is some potential ways of leveraging some domestic and international collaboration to help provide knowledge for policy development. I think Jim rightly emphasized that we need to tackle AMR issues through international systems approaches. And I would go further and say we actually need transdisciplinary systems research approaches between researchers, policymakers, and community groups. So I'm going to tell you about a current international funding opportunity that is seeking to help solve a variety of issues at the nexus of climate, environment, and health. And this opportunity is a collaborative research action through what we call the Belmont Forum. And if I can have the next slide. The Belmont Forum is a consortium of 31 funding agencies representing over 55 countries. We come together around what we call the Belmont Challenge, and I'll get to that in just a second. We work extensively throughout the globe. We've partnered with over 150 resource organizations over the past 10 years to focus mobilizing international resources to address acute issues in global environmental change challenges. So if I can have the next slide. We are guided by what we call the Belmont Challenge, and that is to understand, mitigate, and adapt to global environmental change issues. Our mission really is to lower barriers or remove barriers to international collaboration for groups to come together to focus on accelerating the delivery of environmental research that's needed to help us grow towards sustainability. And we do that by aligning and mobilizing international resources. And as I said before in the first slide, this is a collaborative research action. And for us, that is typically a call for proposals. And we address societally relevant challenges mostly through what we would call transdisciplinary research approaches. And for us, that means that all of our research consortia must contain natural scientists, social scientists, stakeholders, and stakeholders can be anyone from that is relevant to the project being proposed, meaning government organizations, NGOs, community groups, anybody that needs to basically utilize this information in order for decision making. So if I can have the next slide, please. So our mechanism is fairly straightforward, and we've worked on this since about 2012, and we've put out 19 collaborative research actions over that time frame, involving about 2,500 researchers from 70 different countries. Our mechanism is that there is a consortia that writes one single proposal. It goes through one peer review process. And for any researchers out there in the field, they know that that is actually a big step forward. All projects teams, as I said before, must be multilateral. For us, that means there are 3 different countries, so researchers from 3 different countries requesting from at least 3 different funding agencies participating in the call. Each funding organization has an annex that outlines who is eligible to receive their funding, if there are any funding restrictions, and if there are any particular areas of interest. In addition, once we go through this peer review process, the successful proposal consortium members are then directly supported through their national program. So we do not pool any money. There's no commingling of funds, which again has been a big step forward to international collaboration. And again, researchers from countries that are not participating in the call are also welcome, and they can either bring their own funding to the table or provide in-kind contribution. And our big idea here is that we really focus on transdisciplinary teams. So this has got to be natural scientists, social scientists, and external participants that come together to co-produce, co-develop, and co-implement these projects. Have the next slide, please. So we have a new call out on the street for climate, environment, and health. This is the second climate, environment, and health call that we've done. We did one back in 2019. So this, we are going to look at, our goals are to help improve our understanding around climate, environment, and health pathways. And we're hoping to take a One Health approach on this to protect and promote ecological, planetary, and human health in the face of climate changes. We're hoping to integrate knowledge from human health, veterinary medicine, environmental, social sciences, indigenous knowledge, communities, stakeholders, all of those to come together. And really, we are looking to promote and mobilize an inclusive, transdisciplinary funding scheme that's going to support diverse communities, diverse research concepts, and also ecosystems, and to help promote creative, transformative solutions. So if I can have the next slide, please. So we want then to provide information or knowledge, scientific evidence, and tools that are going to help support long-term policy making and decision making. I think we're on the same page as Jim here with the systems level approaches at the intersection of climate, environment, and health. We are particularly interested in focusing on underrepresented communities of different socioeconomic, cultural, gender, and minorities. A lot of marginalized communities are the first ones who are adversely affected by these challenges. In addition, we hope to foster an international collaborative community of practice for this. So if I can have the next slide, we'll go into, we need to skip this one. I apologize, I put in an extra one. Great. So the research themes here are that we are going to look at 3 different research themes, A, B, and C. Theme A is actually one that we are asking all of the projects to have participate in. So we are expecting the projects to have decision science and environmental behavior and implementation ideas, and what that actually means is that we're looking to either develop or utilize knowledge bases to overcome adaptation barriers, things like the adoption of the global action plans on AMR in 2015. There's been a lot of countries that have developed national action plans, but a lot of the challenges that they've had to implement those is the lack of local data to support the decision making, and resource allocation is a barrier to implementation. So those are the kinds of ideas that can come to the table to help move those things forward. We're looking for things where we're trying to understand individual or population level adaptation to climate related threats, so like migration, and the AMR, obviously, community is probably working quite heavily in this, as many of the migrants are affected through their migratory path to through different programs and things. Next slide, please. So the research theme, the second one, is to look at food, environment, and biological security. And so human health and well-being are intimately connected with the environment, both natural and human-built, with animals, wildlife, and livestock. Again, I think some of the things that Jay was talking about with ARGs and soils is a right part for this part of the research themes. We're looking for things that are climate sensitive, from foodborne, waterborne, vector-borne diseases, also things like mental health challenges, including anxiety and stress. So I want to also emphasize that AMR is definitely well within this framework. It's not the only pathway that folks can undertake projects around. In addition, we are looking to understand system level strategies to mitigate or adapt to a lot of the examples that both Jim and Nancy were talking about earlier in their talks. If I can have the next slide. So we're also looking to understand climate risks to ecosystems and populations. And that could be things from a variety of environments, from wildfires, chronic heat exposure, migration of, again, wildlife and insects. So this, again, is going to come straight back to the AMR community, and the interface with domesticated animals and humans. So again, we're really trying to push the system level strategies to evaluate current policies, programs, and implementation. So a lot of the things that Jay was talking about earlier with risk models, using existing data and maps and models, could be used as a way to start to put different parts of the system together between and assess the trade-offs and balances between different potential pathways and policy options. So those are the kinds of approaches that we're looking for. We're also looking to help with understanding health implications, and climate change to improve human and animal health. So we realize this is a very big ask. So if I can have the next slide. Our strategy has been to launch or announce this opportunity on February 15th. What we are going to be doing over the next 5 or 6 months or so between now and when the pre-proposals are due, we are going to be having a series of capacity development through trainings, workshops, office hours, networking, to help the communities come together. We realize that not everybody in environment and climate and in the health world actually work together. So we need to invest in bringing those people together and to help them do, create those networks and come up with ideas. Now, the capacity building activities are informal, they're voluntary, and they're open to everyone. And you don't have to participate in order to put in a proposal. But we have shown in the past that that actually is not a bad idea because it does help you bring things together with your team a little bit better. And so we are working with many resource organizations in order to do this. We have a training schedule to help develop transdisciplinary projects. We're looking at things like providing training on modeling and assessing things like WMO, the World Meteorological Organization, or U.S. government databases. So there are several different of our federal sisters that are working together in order to do this. So can I have the next slide? In addition, we have, as I said before, there's what we call an annex. And each individual funding agency writes one of these things, and it outlines who's eligible, if there's any restriction on the funding and stuff. And so it can be kind of a tricky landscape to navigate. So we have come up with what we call an annex comparison tool. So if you have partners in a couple of different countries, you can actually use the drop-down menus shown there and the little arrows and bring up all of the potential funding partners, and that way you have a one place to understand what it would take to put a team together from those groups. And again, in addition, we have annex workshops that we run with the funding agency representatives. We have our comparison tool. In addition, we put together short video clips on how to do all of this, and then we also have office hours for folks who want to come in and ask questions about what all of this means. Can I have the next slide? So this is where we are so far. As of March of last week, we've had 18 federal and international organizations that have come together to pledge funding for this. We do really want to promote low to middle income country participants as much as we can. So we have a couple of different organizations, the Inter-American Institute for Global Change Research, International Sustainable Development Research Society, and several of the funding agencies within our international landscape and Future Earth are able to actually fund low to middle income country research scientists directly. So we can encourage folks to work within those countries that are, as I think it was Jim that showed, Asia and Africa are highly impacted by this. And so we feel we don't want to support researchers coming into a place, gathering data and leaving. We really want to promote that on the ground community relationship and having the researchers actually work with people in country. So I'm going to have the next slide. So this is where you find all the information. We have a Belmont Forum website, and then we also, I told you about the climate, environment, into health. So my AAAS fellow, Brian Lung there has put together a how-to on how to put all these things together. So we want to bring this to your attention. I think federal agencies are able to partner in this. We've worked quite a few other agencies in the past. Belmont Forum will also accept what we call in-kind contributions, which means that if you're already working on something, you have scientists who are part of a group working on one of these challenges, we can bring them together with potential international researchers to actually leverage the information and the programs that you're working on now. Belmont Forum is all about the idea of leveraging domestic programs that we have investments in through international collaboration around these challenges. So I'll leave it at that. Thank you.
Thank you very much, and thank you to our panel. I'm going to start with one question and then go to Dr. Blazer for a question to start our question section. So I think I'm figuring out that when we talk about environment, it can be multiple different things. It can have different contexts of where it's located. It can be water, air, soil, surfaces, et cetera, how we do that. One of the other questions I have is, this morning, Dr. Phillips referenced, the different concepts of emergence of resistance in the environment, amplification, and transmission of resistance. So my question, and I'll let you, Dr. Garland, start, is you'd talk some about risk assessments, et cetera. Are those different enough that we approach them and consider them differently, or when we're assessing these risks, are those kind of all wadded together?
Yeah, I'd say from the perspective of what I talked about, most of the risk models we're looking at are kind of for existing disease, not necessarily emergence of new disease. Some of the targets that we would use and we are using in that the monitoring program, like colistin, things that are emerging to see, but if they are developing and increasing. And it's somewhat what the metagenomics is for too, to see whether there may be new resistances that are developing. But when you get to the risk models, to that level, and really trying to get it into, say, regulatory or policy decisions, that framework that I mentioned, we're trying to keep the existing QMRA framework and adapt it to AMR, but some of these broader impacts of, say, emergence of new disease, that is one of the adaptations you'd have to make, but not quite sure how to do that yet. So to answer your question, the emergence is important to look at, but probably more as almost a present absence, but not in a quantitative way yet.
Anyone else has comments or not? That's fine. Thank you. All right, Dr. Blazer.
Yes, thank you to the panel for really interesting presentations. I'd like to direct my question to Dr. Chow. The emergence of azole-resistant C. auris and azole-resistant Aspergillus fumigatus is really a terrible event. It's really a very scary event. And, it's hard for me to dissociate it from the intensive use of azoles in agriculture. So I really have two questions. The first is, can you help us develop a scenario of how it moves, how the resistant organisms move potentially from the farm to the hospital? And secondly, as we see with antibacterial resistance, we're using in the non-human space drugs that are useful in human medicine. And the question is, are there, recognizing the utility of these drugs in agriculture, are there reasonable substitutes that are not used in human medicine?
Thanks for those questions. One of the things that, as part of that theory that we had mentioned from Arturo Costa-Duval, just to delineate between the emergence of azole-resistant A. fumigatus and intrinsically resistant C. auris is, they're very different on some level, because A. fumigatus was already out there, it already was on the map, already being able to cause infections, and C. auris was not on the map, and it just emerged and emerged independently. So I think that's sort of one thing for us to consider, that C. auris is special in that way. In terms of transferring from the farm to the hospital, I think it's the standard things that we could think of, that, once it was being able to be entered into the human population, we know that C. auris is a skin colonizer. And it's interesting because, as opposed to other candidas that are mostly gut colonizers, C. auris is predominantly a skin colonizer. So having patients or having people go into the healthcare system, and everything that we've seen about C. auris is that it sets up shop. It's able to persist on surfaces for long periods of time. Some of the studies show that it can persist on surfaces for months, and it's very, it's intolerant to a lot of disinfectants. So I think as a skin colonizer being introduced into the healthcare environment, that's how we've generally seen it. And then the second question, could you expand a little bit more in terms of, you were asking about substitutes? Are there reasonable substitutes?
Yes, but before I expand, I want to go back to the first question and say that the commonality between C. auris and resistant A. fumigatus is natural selection. Darwin was right, and that's the most parsimonious explanation. So the question about but you actually didn't answer how A. fumigatus got to the hospital, resistant A. fumigatus got to the hospital, unless it's the same. Substitutes, the question is, if it's important to have antifungals in agriculture, what kind of agents are out there or that can be developed that would be, let's say, would be too toxic for humans, but okay for plants, and so they're basically not competitive in human medicine.
Yeah. Oh, well, thanks for the A. fumigatus point. There are a lot of groups that have done air sampling in various hospitals, and they're able to find Aspergillus fumigatus. So the idea of that these spores are everywhere and that they're able to go into the healthcare environment as where other environments, I think, is just kind of naturally happening, but it's where it's able to then stay and then, be able to cause infection. So that's one thing. And then in terms of substitutes, I would maybe defer to some other colleagues. I don't know what is exactly out there that could be substitutes to answer your question in that sense. But I do think, just to reemphasize, what we're currently doing to be able to preserve the clinical efficacy of the new antifungals that we do have, because there are so many limited options of antifungals in the pipeline. So being able to think about the considerations of, bacterial, antibiotic, as well as antifungal human considerations into our approval and evaluation processes for agricultural fungicides is very critical. So the group that we've recently formed, we're excited about that.
And Marty, I can just add on the alternatives, there are not really good alternatives, at least in some of the conversations that we've had with EPA, unfortunately. And I think part of it goes to a lot of the conversations that we've had with this group over the years, is that the level of investment that we see on the agricultural side or on the veterinary side in alternatives to antibiotics is not robust. And so I think this is one of the challenges that we face, and it's coming just sharper into focus, that these agents that we've been using for more fungal pathogens have this human analog, and it is a acute challenge.
Thank you.
Thank you, Dr. Lakshminarayan.
Thank you. Thanks for all three of those presentations. So again, a question for Dr. Chow. We've had Dr. Pilcher talk about the fungal, Dr. Chiller talk about the fungal pathogens here, and this temperature issues has come up because we've seen that same picture before. My question is, it's not like we don't have hot places on the planet right now. and places where there's an opportunity for these bugs to evolve even today without necessarily the increase in climate. I do realize that Canada Auris offers a particular example where it shows up in all these different places quite unusually, and therefore there appears to be a climate link. And we've also had Dr. Arturo Casadevall present here as well. That was his paper. But this is something which has always sort of perplexed me. Is it we have hot places. Why haven't these things evolved already?
Well, I think it gets back to everything that we're talking about these environmental changes and rising temperatures. So you're right, we have hot places. When we look at C. auris, where it emerged, it is often in these areas at first that were in these very, hot coastal areas, many of which somewhat near the equator, with the exception of South Africa for some of these first outbreaks. But I think it's just what we're seeing as a matter of time. So as we're seeing these rising temperatures, it takes time for things to be able to evolve and adapt so that, it's the thermotolerance is enough. So I think it's just time.
Thank you. This question is for either Dr. Stack or Dr. Chow, and it goes back to really based on Dr. Blazer's question. And so when we think about the development of, say, a new antifungal, right, it could cost a drug company hundreds of $millions or $1 billion. And when you look at the amount of profit based on treating humans versus allowing it for agriculture, I'm sure the agricultural profit is way up here versus what it is with the much smaller group of humans that would be treated with the drug. So in order to think about trying to preserve it for the human population, is there some way that we've thought about incentivizing, so not to, say, for example, you were talking about olorofen, that immediately comes out for humans and they've already applied for their agricultural license for that. Is there any way that in some type of a 5-year incentive or whatever that we subsidize so that they don't yet go into the agricultural field in order to keep that initial preservation for human treatment? I don't know what the answer is, but if I owned a lot of stock in that company, I would want them to be selling it, right, to the industry that's going to make the most profit for the drug company. And I think maybe that's part of the problem. How can we incentivize them not to do, at least for a certain period of time?
Take it.
Sorry, that was a tough one.
I think there are a lot of different issues embedded in your question. So first of all, if you look at the structure of the industry and the strategies that they use. So a long time ago, decades ago, the fungicides that were used in the plant sector were very general in mechanism of action. And so the consequence of that, of course, is you have a lot of non-target effects. And so they were driven by regulation to develop products that had more and more specific modes of action, which increases the likelihood of developing resistance to them. The other is, if you go back to the early 1990s, we probably had 15 companies, 16 companies globally making fungicides for agricultural applications. I think we're down to 4. And so that market incentive's not there. They need big selling products. And I think the life cycle is not anticipated to be very long at all. So there isn't a great deal of incentive. We've lost a lot of products. There are literally no products in some application areas on the plant health side. There are just literally no products left. And there's just, the profit margin is too small to develop a product. So I think we're going to have to make some tough decisions. I mean, to me, it's just not rational that anybody can develop the chemistries for whatever application they can find a market for. I think we're going to have some kind of a structure in place that says use these in the public health sector, use these in the ag sector. If we don't do that, this problem continues.
Ms. Ewell, you have your hand up, please.
Yeah, I think I just want to comment on your question there, in the sense that your question is exactly the kind of thinking that these calls for proposals are trying to get at. We're looking at what are trade-offs and balances. How do you incorporate financial models and balance that with human health, environmental health? All of these kinds of approaches are necessary, and as you can see, they're tricky questions. And I think Jim's point about taking things from a systems approach is critical, because you can't make one decision in one sector without it affecting or potentially jeopardizing something in another. So if we want responsible stewardship and sustainability moving forward, these are exactly the kinds of research questions that the group should be working on. And you should have the federal resources and private resources to come together to look at exactly those kinds of things. Thank you.
Mr. Craig.
Thank you. So a quick comment to follow up on that question, because I think it's an important one, because I think this Alora film issue is a significant one that we need to recognize. And I'll just flag for you that actual antifungal agent was the company that John Rex, who used to be a part of PACCARB, is actually bringing to market. So we do have a PACCARB connection to that antifungal agent. And I will note that, I think as Christine's commented, it's a challenge because there's also two different regulatory cycles in play here. There is the EPA pesticide regulatory cycle, which I will say is very short relative to the FDA regulatory cycle, which is very long, which makes sense because we're putting one of them in our bodies. But that difference in cycle creates the opportunity for, when research gets published, say related to the development of a new drug or what's going to come up, that a pesticide company could identify a new form of action and market and adapt a pesticide and actually bring it to market faster than that FDA approval could actually happen. And we could also be in the situation where we could be investing hundreds of millions of dollars, not just of the company's money, but potentially of public money, whether it's from the US government or CARB-X or some other organization that's contributing resources, and then undermine our own investment with the approval of that pesticide. So there's some really challenging dimensions of this that we do need to sort of, bring to the table and talk about. And I think thanks, Nancy, for bringing up that EPA group, because I think that is going to be a group that has to really wrestle with some of these things and consider how do we address some of these pieces. So we just want to comment on that. And then one question for Nancy, because I think one of the things that I think would be good for the committee to hear is all of the work that we are doing to improve detection of Aspergillus fumigatus with the AR Lab network and some of the work that we're doing. And I think that you're leading on whole genome sequencing and trying to also understand some of these relationships. Can you talk about some of that?
Yeah, certainly. So excitingly, with the AR Lab network, we've been working with departments of Tennessee as well as Maryland to collect as many clinical isolates of A. fumigatus and be able to screen for azole resistance. And so that's, establishing some form of laboratory surveillance. And it's all about surveillance. I mean, everything that we've been talking about in the last panel here, this holistic One Health surveillance approach. And I would just add that environmental sampling, it's really hard. Like when we start thinking about, sampling for Aspergillus fumigatus, I mean, Dr. Garland, you talked about the standardization of specimen collection. Also, the analysis is really hard. We hear a lot about metagenomics, but if you really look into some of these efforts, they're not doing metagenomics quite yet. It's really actually PCR-based methods, but trying to do metagenomics because there's so much that's needing to be done in terms of being able to specifically say, this is a species, this is a strain with metagenomics. So we're trying very much to advance that. And one of the things that we and other colleagues have found is that metagenomics, surprise, surprise, is very different for fungi than it is for bacteria. It's very hard to get enough sequencing depth, what they call that coverage, to be able to detect a fungus metagenomically in a sample. So those are some of the things that we're trying to advance.
So I'll finish up with one more question here, and if anyone has more. Oh, okay, yeah, go right ahead.
Thanks, everybody. This is always fascinating to learn stuff that I never even thought I didn't even know. I didn't know some of the stuff that you guys bring up. Now, obviously, Candida auris and Aspergillus fumigatus I got as a pediatric infectious disease doctor. But if I'm sitting out there today and I'm watching wherever you can watch from today, why do I care if I'm a layperson about any, Aspergillus fumigatus or Candida auris? I mean, for many people in this country, Candida auris became something maybe new on CNN this past week with the publication. COVID was nothing until it started killing a bunch of people, right? And we started seeing that it mattered. And now we're like, oh my goodness, I guess I got to worry about this virus. So while all of us in this room can feel it and know it because we can see what will happen next, whether it's, the agricultural piece of this or with the, what water, and we know it has all the influences and could impact our health. I mean, unless I see strep throat, which has been something big recently with group A strep and invasive, right? People don't know what this means to them. So how do we I think it's easier for Candida auris and maybe Aspergillus. So most of our friends and family have never had an Aspergillus infection or a Candida infection. How do we make them care? How do we make all of this matter? And I'll leave that open for everybody to kind of consider because I think that's one of our biggest challenges going forward.
Do you want me to, well, from the CDC perspective, do you want me to? Okay. Yeah, well, that's a wonderful question. I think we have to feel how we can relate to being in the situation where you are at risk for an infection, like an invasive mold infection. And we know that people who are immunocompromised, but that's easy to say that, but certain medications that people take, these immune modulators, TNF inhibitors, a lot of people take that for various reasons. And we have family members that take it for, sometimes prolonged periods of time. We have many, many family members that are living longer now and going through a variety of surgeries, case in point, my father recently. And these are things where you're exposed to the healthcare environment time and time again. And these are real infections that we see high mortality rates. I mean, 40% or more for invasive Aspergillus fumigatus. So I think just being able to know that this represents patient populations that we all know people like this.
And if I could just jump in there for a sec. I mean, I think Nancy's right. I think the other piece of this is something that Ramadan mentioned earlier is how do we make sure that we are connecting the dots for things that are related that people do understand. And Katie Capers on the comm side presented at the last meeting about some of these pieces, but how do you draw a connection and make it clear to folks that, you know what cancer is, but do you actually know that people are surviving their cancer and dying of a resistant infection? How do you draw some of these connections in ways to some of these communities where we have a better understanding and there is maybe a greater concern with some of those things, but a lack of recognition or a lack of understanding with antimicrobial resistance? And I think we can do that, but I think we have to be a lot more thoughtful about it and make some of those connections in ways that we haven't been able to before.
And I guess from an EPA perspective, it's kind of through the lens of the regulations. I mean, people want to know, is the water safe to drink? Is it safe to recreate in? And so whether a lot of that decision happens at the state level following federal guidance, they want to know that. And so what we're trying to do is give them, and traditionally we have not incorporated AMR into that risk assessment, and we're trying to incorporate it to be able to answer that question.
Ms. Yuel?
Yeah, just my background comes from the climate group. So we've learned a few lessons around creative messaging to populations. And I think, a lot of the mistakes that were made early on in some of the things that we did was the lack of engaging with people who are actually affected, putting things out to populations without really understanding and talking with the people who are part of that community is important. If you're going to have consistent, strong messaging, it does need to be tailored toward the community that you're targeting. And there's no better way of doing that than actually working with those people on the ground to understand what they want, what they need from their perspective. You can have a lot of people sitting around these types of committees that all are well versed in a lot of this stuff, but what happens is it becomes too diluted and too overbearing for, I think, a lot of folks. So I think it's really important to work with not only federal agencies, state agencies, but NGOs, to work with community groups for these kinds of campaigns. And that goes for any kind of, I think, science-based decision-making, is that people need to understand and be familiar with and have those messages resonate with them so that they can actually take this information and make their own decisions. Thank you.
Dr. Jenks?
Thank you. And my question is really for Dr. Garland, and the statement you made that the good quality water, there's an association that there are fewer ARGs associated with good quality water. And this kind of ties to your last statement about the risk assessment. And of course, people want to have water that's safe to drink, safe to recreate in. I guess what I want to understand is, are we trying to figure out how much of this is the anthropogenic drivers of antimicrobial resistance and separate out from the natural sources of AMR and ARGs? I mean, I'm sure there's a very precise definition of what safe or good quality water is, but not all water in the world probably falls into that category. So how does this kind of break out when we know that ARGs are out there already? And that's not quite what we're concerned about. We're concerned about the risk factor.
I think this gets into, well, there are indicators, right, that we have and threshold levels for whether we're going to accept, whether we're going to close a beach, for example, right? So there's a threshold of fecal indicators that, based on studies of kind of epidemiology and what the disease rates are, we've defined what a level is. Now, what is that level for AMR? That's what we don't know yet, right? And I think that's what we're trying to get at. So developing indicators is one. But the primary question, when I said the ecological condition of a water is impaired, and that's associated with higher levels of ARG, that relationship was clear and maybe not that surprising, right? The real question is from a mechanistic point of view, is trying to understand, number one, is it just a matter of what contributions or what impacts that stream has received? So therefore, there's higher level of AMR risk because it's receiving human wastewater, agricultural wastewater, whatever. That's more traditional approach here. But the other component of this, and really thinking about the environment, is the environmental condition leading to the increased prevalence of ARG, not because it's just the die-off or attenuation of the input, but rather the condition, the ecological condition, is leading to higher levels. And that's what we're trying to get at with some of the relationships that we hope to gain from this study, where we're looking at ARG levels versus, say, fecal indicator levels and understanding what the association is. But I think the smoking gun for that kind of relationship would be where a site where we don't see fecal indicators. We don't see high levels of that, but we do see high levels of ARG. And maybe that's because the overall ecological condition is poor for other stress reasons, and therefore promoting the occurrence of the ARGs. Now, is that from natural sources? Probably not, because again, with the study design, you have the reference sites where you can kind of try to tease that out or compare that, sites that really are not stressed at all, the least impacted. Hopefully that helps.
Dr. Dodds, Ashley?
Thanks. And I'm going to a little bit jump back to Jason's question, but some things just really struck me with that, and also ties into Dr. Fukada's talk from this morning when you brought up the example of we need the message that's going to make people want to act like they do for pollution and eliminate use of fossil fuels as much as possible. I think that's part of the problem. I think, Jason, we don't need to make patients worry about Candida auris and Aspergillus. I think we need to find a way to speak to everyone about AMR and then bundle these efforts together. And I think that is the way that we can tie in our colleagues doing environmental work, but makes it so hard. And it also ties into our report for tomorrow, where we talk about communication being so important. But I guess, and I don't really truly have a question, but I just wonder if there aren't some best practices we can learn. We had a great successful campaign. You cannot buy a piece of chicken with antibiotics in it anymore. It is on the door of every restaurant you go into that they serve antibiotic-free chicken. But I feel like we almost lost the opportunity to capitalize on that being an AMR intervention. And it somehow came about, not giving your kids antibiotics and chicken. Maybe I'm just biased because I spend a lot of time with kids who eat chicken nuggets. But, and I don't know if anybody, and maybe others on the council have it, but it's such an opportunity for us to combine these efforts and maybe not be a splitter, but be a lumper when it comes to how we approach AMR across all these various areas. I don't know if any of you have any creative ideas on how to do that, or any CDC work, Michael, that might be headed that way, but.
I mean, I think on the communication side, as Katie talked about, we are trying to think through the messages, and I would agree that, we do sometimes focus, and the news does this a lot, of course, as we saw this week, on a single pathogen. And we get so fixated on the pathogen du jour. And, today, this week, it might be Candida auris. We're going to hear from Dr. Walters about the eye outbreak, which was Pseudomonas. And then we've had MRSA, and, we had a Shigella, report recently as well. And I think you're right that part of it is we need to make the thread and communicate more effectively that, while there are these individual pathogens, what we're ultimately talking about is that greater issue, and how do we do that more effectively? And then also, as Katie talked about last time, is how do we personalize it so that there is some self-efficacy of what can that person do to protect themselves or their family? What are the steps that they can take for some of those things? And then, also make it clear that, it's not all on them, right? That there are efforts for the government to take action, for groups to take action, and for everybody has a role to play, essentially. And how do we do that more effectively? So it's hard. And I think that's the challenge, of course, with all of these environmental topics are so humbling, because you see the literal global complexity tying every different piece together. And then how do you sort of communicate about that effectively so it's not too overwhelming, and that it actually, matters to somebody so that they can do something that helps themselves.
I was just going to say, I don't know if this answers your question, probably not, but, this morning when I asked the question to Dr. Pruden about the structure of that UNEP report about the value chains, right? I mean, we value healthcare, we value ag, we value, what was the third component? But, there's things we value. What we don't value is clean water, right? So the message is, you want clean water? Do you really value it? Because it's going to take an investment to do that. And so I don't know how that's a hard thing to communicate. But the other thing that I would say, maybe, and this is building off what Dr. Fukada said this morning too, about linking to other initiatives. The initiative that is looming, and it's within EPA, within the water, within the U.S., is PFAS. and perfluorinated compounds. I mean, there's now a action level out there that people are going to have to meet. And by meeting that, they're probably going to have to treat to a level that certainly might improve or reduce the risk of AMR, particularly when it comes to, say, biosolids. I mean, what you're going to have to do in terms of thermal destruction, even of that material, to have safe disposal and use of biosolids, probably is going to help solve your AMR problem to a certain extent too. So maybe and I don't know, maybe is it easier to communicate chemical risk than AMR risk?
Ms. Yule?
I just want to kind of reiterate the messaging here, having it be broad enough, but have it linked to real world experiences. And I would really push to work with communication specialists. Scientists, you get them in a room and we go down a rabbit hole fast. And a lot of the things that, the idea behind being a lumper versus a splitter, we can have our academic discussions around that. But I think when you're dealing with messaging, it has to be crystal clear, it has to be relevant, and it needs to be able to be personal so that people can actually see themselves in it. In addition, it also has to provide them with potential ideas for action. The worst thing I think people can do when we're talking about these kinds of things is to raise the red flag and then just say, this is all horrible, and that never provide any kind of guidance on what you as an individual can do for this. And I've been in this business for quite a while, and the science communication still needs to go a long way. And I really do want to emphasize that as scientists, we have our own expertise. Well, there's people out there who do communications as their expertise. And I really think it's time for us to come together and embrace those two groups to be as most effective as possible. Thank you.
Thank you. Dr. Simpson.
Yeah, thank you. And I think this point has kind of already been made, but I really do want to emphasize in response to your point, Libby, the lumping versus splitting, and what Dr. Fukada said earlier about sort of using opportunities presented by other agendas and sort of fitting AMR, instead of trying to get people to understand what AMR is or be afraid of this pathogen du jour. I think Ramanan had said earlier this idea of horizontalization and connecting AMR into bigger, more sort of universal human concerns like child health, the needs of the aging population, vulnerable populations. I think finding ways to align the issues of AMR, because they're all intertwined with these larger sort of social movements in various spaces, may be a way to think about it. So just emphasizing that really stuck with me from this morning. I'm glad we're sort of converging back on it. Thank you.
Thank you. Dr. Musmar tells me I'm supposed to close out, but I got 3 minutes and the mic. Dr. Stack, you brought it right down to the gist of it about who gets to use what, I think. And so having started hauling wheat at 14 years old to the elevator and remembering the wet years with the rust coming through, et cetera, could you put into perspective for the council what it would cost to apply something for wheat rust or an antifungal to a crop in relation to the profit for an acre, to put into perspective the cost it takes to actually apply those?
Yeah, so I'm probably out of date on this, but for, say, the strobilurins, that class, last I recall, depending upon the number of things, but somewhere between $25 and $40 an acre. And that, I don't know what that sounds like, but when you look at the profit margin on a grain crop, that's eating into it quite a bit. And oftentimes it's more than you can justify the application. And so it's a very hard decision for a grower to make.
Yeah, I just thought that's important that we can't just, it's the same thing as antimicrobials and food animals. You don't just do it without a real economic hit when you do that. And same on the family farm back home, and sometimes it takes more than one, right? If you're going to try to do it.
Absolutely. So it depends on the commodity. So for example, potato production in South America, it's 14 or 15 fungicide applications per production cycle. So almost every week once the foliage really fold out. Whereas with wheat, you don't want to put any on because the profit margin is so small on that production. I wanted to make just a color. So the resistance problem is not the only health challenge when it comes to agricultural chemicals. Just chronic exposure is a major health issue, especially in developing countries. And the indiscriminate use is a major issue, and it's not just in developing countries. We had a new disease introduced into North America on maize, on corn, in 2014, and it took them about 18 months or so to figure out what it was. And in that interim, the farmers are spraying fungicides for a bacterial disease because they needed to do something. And until it got sorted out what the actual cause was, it wasn't clear that they were doing that for nothing. They were actually losing money on the application. They were applying the product that cost them money and not getting any benefit from it. So that goes on all the time, where you get misapplication. It's analogous to prescribing an antibiotic for a viral throat infection. That happens in agriculture too.
Thank you very much. Well, my watch shows directly on time, Dr. Musmar, and thank you to the panel for great presentations, and thank you to the council for your discussion.
Thank you for staying on time, Dr. All right, folks, we're just going to take a 5-minute stretch break as we prepare for our public comment. We have a few people in person, but the rest are virtual. And then for everybody at the table, please prepare your notes for the round robin to take place afterwards. Thank you. 5-minute stretch. Don't go anywhere. Come back.
Produced by the U.S. Department of Health and Human Services.